At the very onset, I would like to welcome all of you to my class today, which we will be mainly covering on the microbiological assay. This is a part of pharmaceutical microbiology, which is a curriculum as recommended by Pharmacy Council of India, PCI, as well as followed across the country in various universities, including Dr. APJ Abdul Kalam Technical University. Now, before I proceed to describe what was the logic or principle behind this antibiotic assay, I would like to quickly recapitulate what basically antibiotic was. Antibiotic were basically those compounds which were synthesized by microorganisms in very large quantity, used to inhibit other microorganisms in very minute quantity. Now, there are a large number of microorganisms which are producing various antibiotics as their metabolites, and they are generally produced in very large quantity. Say for example, let us talk of a very common antibiotic which we are calling as streptomycin. Streptomycin is produced by a microorganism called Streptomyces griseus, and this organism Streptomyces griseus is producing streptomycin in very large quantity. At the same time, this streptomycin has been found to be having a very good action against other microorganisms belonging to the gram-negative species. In fact, this inhibitory capacity we measure, we estimate whenever we are going for the microbiological assay. Now, in the previous class, we have studied about the antibiotic assay by cup plate method, and as I have told you earlier that the assay of antibiotic can be done in two methods mainly. One is called the cup plate method, where we are estimating the zone of inhibition of the antibiotic which has been producing the zone of inhibition, we are measuring it. So, we are taking the help of solid nutrient agar media, and in case of the other method which is called the turbidimetric method, where we are utilizing a liquid media, and in this particular liquid media we are measuring the turbidity. But the principle remains the same. What is the principle? Here in cup plate method as well as in turbidimetric method, we are estimating the degree of inhibition. The degree of inhibition being produced by the antibiotic. So, the fundamental difference between these two methods is in this case of a cup plate method, we are measuring what we are calling as zone of inhibition, whereas in turbidimetric method, as the name implicates, that is metric means measurement, turbid means turbidity. So, we are estimating the turbidity in case of turbidimetric method, whereas in the case of couplet method we are measuring the zone of inhibition. Then in the previous class we have discussed about in details the one level factorial assay. In today's class we will be continuing with that of what we are calling as the two level factorial assay. We are calling as the two level factorial assay because we are taking two potencies of the standard which we are calling as S1 and S2, and we are calling for unknown we are calling as U1 and U2. And with the help of measurement of the zone of inhibition, we will be calculating the two level factorial assay. So, let me explain in a bit detail how we are carrying out the two level assay. First of all, we have to prepare parallel dilutions of two levels of standard S1 and S2. S1 the lower level, S2 being the higher level, and we take two levels of the unknown of the samples as U1 and U2. In fact, that is the reason why we are calling this method as the two level factorial assay of an antibiotic. And each of these four plates fill each of its four cylinders with different test dilutions, that is, we are filling up alternate with S1 and S2, U1 and U2. And then after this nutrient agar plate flooded with these microorganisms and then putting this antibiotic in the respective cylinders, followed by the process of incubation, will lead to development of zone of inhibition. And then we have to estimate, we have to measure this zone of inhibition as produced by the two levels of standard S1 and S2 and two levels of unknown U1 and U2. Let us see what formula we adopt. We are measuring what we are calling as potency. Potency we want to measure. So, let us see the formula first of all. Percentage potency is defined as anti-log of A plus 2 log I. I am repeating once again. Percentage potency is equal to anti-log of A plus 2 log I. So, first of all we need to define what is I, what is A. I is nothing but the ratio of the dilutions, that is the ratio of the concentrations or the dilutions which we have taken for our antibiotics. So, I is nothing but the ratio of dilutions, and on the other hand the value of A is equal to U1 plus U2 minus S1 plus S2 divided by U1 plus U2 plus S1 minus S2. Whereas U1 and U2 are nothing but the sum of the zone diameter of unknown high and unknown low. S1 and S2 are the sum of zone diameter of standard high and standard low, because you have taken two levels of standard which were S1 and S2. You have also taken two level of sample or unknown. U1 and U2. So, U1 and U2 are the zone diameter of U stands for unknown, unknown high and unknown low. S1 and S2 are the zone diameter of standard high and standard low. Therefore, you put the value of the zone diameter to be here. So, U1 plus U2 minus S1 plus S2, U1 plus U2 plus S1 minus S2. This is the formula which you have to adopt to determine what is called antilog of A. The value will come up from here, plus 2 log I. I is nothing but the ratio of dilutions. So, whatever value you are getting is known as percentage potency. So, with the help of the formula written, you will be in a position to estimate the value of percentage potency, and the potency which is assumed multiplied by this percentage potency divided by 100 will lead to the development of or the calculated potency after the process of experimentation. Now, we are starting with, as I have told you, that this antibiotic assay can be carried out either by turbidimetric method or by tube dilution, either by turbidimetric method, common another name is tube dilution, or by couplet method. Now, this particular table shows you the list of antibiotics along with the methods which are recommended as per Indian pharmacopoeia. Now, amikacin is estimated by the turbidimetric method, erythromycin is estimated by couplet method, penicillin G estimated by couplet method, oxytetracycline estimated by tube dilution method, amphotericin B estimated by couplet method, and chlortetracycline estimated by tube dilution method. These methods are specified by Indian pharmacopoeia. Now, there are certain logic by virtue of which these methods are recommended by IP. If the antibiotics are perfectly soluble in water and they are not leaving any particulate impurity, we will preferably go for turbidimetric method, else we will go for the couplet method. If solid impurities or if particulate impurities are there whenever we are assaying an antibiotic, by turbidimetric method, there is a chance of confusion that this turbidity which is being developed after the process of incubation, whether it is due to the particulate impurities or due to the presence of the microorganisms. Now, let us come to what are the organisms which are specified or recommended by Indian Pharmacopoeia. IP recommends amikacin to be estimated or evaluated or assayed by Staphylococcus aureus ATCC, American Type Culture Collection 29737. Erythromycin is estimated by the cup plate method, where the microorganism recommended is Micrococcus luteus, American Type Culture Collection 9341. Similarly, penicillin G estimated again by cup plate method, where the microorganisms recommended by IP is S. aureus ATCC 29737. Oxytetracycline, on the other hand, is measured by this turbidimetric or tube assay method, where the organism is Bacillus cereus variety mycoids with a strain number ATCC 11778. Amphotericin B, on the other hand, it is measured by cup plate method, where the organism recommended by IP is Saccharomyces cerevisiae NCTC 10716. National Collection of Type Culture NCTC. Similarly, in case of chlortetracycline, which can be assayed by this tube assay technique with the help of organism Staphylococcus aureus NCTC 7447. Now, these organisms are absolutely specified by Indian Pharmacopoeia. Not only these organisms, the strain numbers are standardized strains. That is the reason why they have mentioned ATCC or NCTC strain numbers. You have to be very specific as far as the process is concerned, as far as the microorganisms are concerned. Now, this method of turbidimetric method, what we have to do is, in the same method as we have done earlier, we have to prepare 5 different concentration of standard solutions. 5 different concentration of standard solutions. Now, whenever you are preparing 5 different concentrations, we have to take the pure drug. That is the reason why we are telling as that to be as the standard solutions. There should not be presence of any impurity. What we are calling as, say for example, we are wanting to test erythromycin, we have to take erythromycin reference standard. Now, these concentrations are such that S1 is the lowest concentration, S5 is the highest concentration. S1 is the lowest concentration, S5 is the highest concentration, and the ratio of the dilutions generally is 4:5. That is, we have to prepare 5 different concentrations of the standard in the dilution factor of 4:5, and in this 4:5, S1 is the lowest concentration, S5 is the highest concentration for the standard solutions. And these solutions in quantity of 1 ml is to be added in sterilized test tubes in duplicate. They are to be added in a volume of 1 ml in sterilized test tubes in duplicate. Now, you take the sample solution whose concentration is unknown to you. You take sample solution whose concentration you want to measure. So, what you do is, in duplicate tubes you take 1 ml of S1, S2, S3, S4 and S5, each test tube containing 1 ml, it should be in duplicate, and you take 2 additional tubes where you add the sample, that too in 1 ml. Now, question is, what is the dilution of the sample which you have to add in those 2 test tubes? Now, what you have to do is, you are having a labeled claim in the package or in the strip of the antibiotic. Taking that to be 100% true, you prepare a solution which is equivalent to the S3 concentration of the, which is equivalent to S3 concentration of the standard solutions. Now, you are having total 10 test tubes where you are having 1 ml of standard solutions, either S1 or S2 or S3 or S4 or S5. Now, we have taken 10 test tubes containing 1 ml of either S1 or S2 or S3 or S4 or S5. You take additional 2 test tubes where you have to add 1 ml of the unknown sample solutions. Now, this unknown sample solution has to be prepared as far as the labeled claim, that is, whatever is claimed in their package, you have to dilute it in such a manner that it is coming closer to the concentration S3 of the standard. Now, you take 9 ml of nutrient medium, basically nutrient broth, which you sterilize in an autoclave initially. This nutrient broth is containing all the essential nutrition which the microorganism require for its growth, but it needs to be sterile, so that this nutrient broth do not contain any extraneous microorganisms. Now, after you are sterilizing this nutrient medium, I have shown you one slide earlier, there was a list of microorganisms against various antibiotics. Depending on the antibiotic which you are testing, you select a particular microorganism and you inoculate that organism into the nutrient media which you have sterilized. A question may come up in your mind, sir, we are adding microorganisms, then why are we going for sterilization? We are going for sterilization because the nutrient media must be free from any microbial contamination, and the microbes which you are adding later on, that only should be present, and we will evaluate the effectivity of the antibiotic in inhibiting the microorganisms. Now, we are having 3 control tubes. One is known as culture control, which is containing the inoculated culture media. Next, we are having an identical set, but treated with 0.5 ml formaldehyde solution, which you are calling as a blank. And the third one is uninoculated culture media. Ideally, in this uninoculated culture media, it should be absolutely clear, because you are not adding any microorganism, that is the reason why uninoculated, so there should not be any growth. If there is accidental growth, it means the media was not properly sterilized before adding the sensitive culture. The first one with inoculated culture media, that is this culture control tube, it ideally should be turbid, because you are adding the culture microorganism, it should grow, the nutrition is fully furnished with the nutrition, the microorganism should grow and turbidity should result. If accidentally there is no turbidity in this culture control tube, it means the nutrition was not properly given in the nutritional media, that is the nutrient media which you have prepared was not complete. Therefore, ideally this should be turbid, this should be clear, and we are adding a 0.5 ml formaldehyde solution as a blank, so that this formaldehyde will arrest any further growth of bacteria. Then all these test tubes are placed in an incubator at 37 degree for 3 to 4 hours, where we are adding 0.5 ml formaldehyde solution to prevent the growth. Then you measure the absorbance at 530 nanometer for each of the test tubes, keeping blank. Initially you set the spectrophotometer reading adjusted at 530 nanometer with the blank, and then one by one you measure the value of absorbance for S1 to S5, and also you measure for S sample. Now you will be getting a graph. The graph is obtained or plotted where we are taking concentration versus absorbance, and you have to plot this graph with the standard antibiotic solution. So again it will be a straight line, and then you determine the absorbance of the sample or the unknown, you extrapolate it on the graph, and you will be getting the concentration of the unknown sample. Now if it so happens that the absorbance for S1 is A, S2B, S3C, S4D, S5D, ideally it should be a straight line. Now then, if it is not coming in a straight line, you simply adopt this formula where L is equal to 3a plus 2b plus c minus e by 5 and H is equal to 3e plus 2d plus c minus a by 5, where L is the calculated absorbance for the lowest concentration, H is calculated absorbance for the highest concentration. And then you have to plot the standard response curve, and then whenever you then finally you multiply by the appropriate dilution factor to get the antibiotic concentration of the sample. So, you have to first plot a response curve, average the response of the sample and read from the standard response line whatever concentrations you are getting, multiply it by the dilution factor to get the actual concentration of the unknown sample. This is the spectrophotometer which we used for our study, for our determination of absorbance. Initially adjust the value with the help of a blank and then determine one by one the concentrations of absorbance of S1 to S5 and finally the absorbance for the unknown solutions. Now, whenever we are determining, it officially says the value should lie between 95% and 105%. The value should lie between 95 to 100%. So, degree of precision is the minimum acceptable for determining that the final product complies with the official requirement. The fiducial limits for error of estimated potency should not be less than 95% or more than 105%, which is stated in the monograph. This is the pictorial representation of the process which we just now discussed. Advantages: it is having a shorter incubation period, so you are in a position to tell the results within a maximum period of 3 to 4 hours. Disadvantage: as I have told you that if there is any solvent inhibitory substance or if there is any particulate impurity, the process gets very difficult to predict whether the turbidity is due to the particulate matter or it is due to the presence of the microorganism. Therefore, this method is not recommended for those preparations which are generally turbid by themselves or they prepare the media cloudy. This is a topic which you need to cover from again Indian pharmacopoeia. So, both the cup plate method and turbidimetric method, these two topics of antibiotic assay, you have to refer to IP. Either you can go for 2007 IP or you can go for 1996 IP. With this, we are coming to the end of the class today. So, I hope you have understood the objective of the antibiotic assay by cup plate and turbidimetric method. Okay, thank you very much.